Hi, so today we're gonna talk about Krebs cycle, but first I will share to you on how to remember names of intermediates. These are the intermediates included in Krebs cycle. So there are easy way to remember all of these intermediate. All you guys need to do is remember this sentence. Can I keep selling substances for money, officer? Can I keep selling substances for money, officer? As you can see here, the first alphabet, C, citrate, I, isocitrate, K, ketoglutarate, alpha ketoglutarate, S, succinyl-CoA, S, succinate, F, fumarate, M, malate, O, oxaloacetate. So, ramai yang tanya nanti macam mana nak bezakan antara dua S ni. So, as you can see here, ATE, late. Late always later, right? So, succinyl-CoA always come first before succinate. Okay, for energy change, you have to remember 2-NADH occurs where carbon dioxide is released. Here, carbon dioxide is released, carbon dioxide is released, so they have energy release here. Then 1-NADH at final step here, and for 1-FADH2 at succinate to fumarate here. So you have to remember this, where carbon dioxide is released, final step, and succinate to fumarate. Now I will explain if the question asks for a Krebs cycle. So usually the Krebs cycle occurs at mitochondrial matrix. Okay, first acetyl-CoA will join with oxaloacetate to form citrate. This reaction is irreversible. So the next step, citrate undergo isomerization. It removes H2O to form isocitrate. Isocitrate undergo oxidative decarbonylation. It reduces NAD+ to NADH and H+, and it removes 1 carbon dioxide. Okay, alpha ketoglutarate will undergo oxidative decarbonylation. It will remove 1 carbon dioxide and NADP+ to NADH and H+. So it will form succinyl-CoA. CoA will join with succinyl-CoA and it will form high energy. High energy between succinyl-CoA and CoA will cleave. It will release energy and it used to bind phosphate group with CDP to form GTP. GTP transfer its phosphate group to ADP to form ATP. Succinate will undergo oxidation to form fumarate. It removes 2 H atoms to reduce FAD to FADH2. And then fumarate will undergo hydration, which is add 1 H2O to convert to malate. Malate is oxidized and it removes 2 H atoms to reduce NAD+ to NADH and H+. Oxaloacetate will regenerate and combine with another acetyl-CoA, and the cycle is continuous.
So today I'm going to explain about the electron transport chain and chemiosmosis. There are 6 components that involved in ETC. First, NADH dehydrogenase, which is complex 1. Yang second, succinate dehydrogenase, which is complex 2. Third, coenzyme Q or ubiquinone. Fourth, cytochrome C reductase, which is complex 3. and fifth cytochrome C, and the last one is the cytochrome C oxidase, which is complex 4. Next, I will explain about the ETC. NADH and FADH2 was produced during the link reaction, glycolysis, and Krebs cycle. So, electrons in the NADH and FADH2 must be transferred to the oxygen in order to form water. So, first, NADH, complex 1, which is NADH dehydrogenase, is more electronegative than the NADH. So, electron in the NADH must be transferred to complex 1. So, complex 1 will reduce and the NADH will oxidize to NAD+. Next, electrons from the complex 1 will transfer to the ubiquinone or coenzyme Q. So, as ubiquinone move freely in the inner mitochondrial membrane, so it will pass the electrons from the complex 1 to the complex 3. Then, complex 3 will transfer the electrons to the cytochrome C, and the electrons will transfer to the complex 4. And then, as the electron moves, it will move to the last electron acceptor, which is oxygen. In order to form water, oxygen have to receive 2 electrons, but from the NADH, it will only receive 1 electron. So, another 1 electron will receive from the FADH2. So, FADH2 will transfer the electron to complex 2. Why not complex 1? This is because complex 1 is less electronegative than the FADH2, while the complex 2 is more electronegative than FADH2. So, electron from the FADH2 will transfer to the complex 2. So, the proses punya laluan sama seperti dengan NADH. So, dia akan lalu cytochrome 2 and then ubiquinone, then transfer electron to the complex 3, then cytochrome C, and then complex 4, and the last one is the electron acceptor, which is O2. So, O2, in order to form water, 2 electron plus dengan 2H plus dengan half O2, so become water. So, water ni dia akan keluar as a water vapor during breathing. As protein complex receive and donate electrons, they will pump H plus from the mitochondrial matrix to the intermembrane space. For 1 molecule of NADH, it will generate 3 protein complex in order to pump H plus that occurs at the complex 1, complex 3, and complex 4. While for the FADH2, it will generate only 2 proton in only 2 protein complex in order to pump H plus, which is complex 3 and complex 4. Concentration in H plus in the intermembrane space is higher compared to the mitochondrial matrix because they are complex 1, complex 2, and complex 3 pump H plus to the intermembrane space. So, it will create a concentration proton gradient. So, proton gradients will cause the H plus to flow back into the mitochondrial matrix, but it will flow back through the ATP synthase, not the inner mitochondrial membrane, because the inner mitochondrial membrane is impermeable to the H plus. So, it will move to the ATP synthase. So, bila the flow back into the mitochondrial matrix through ATP synthase will activate and phosphorylate ADP plus phosphate to become ATP. So, that's the process of chemiosmosis.
So, before we go through process fermentation, kita akan melalui proses glycolysis dahulu. So, proses glycolysis ni apa? Macam yang korang dah tahu, proses glycolysis ni ialah proses di mana glukos dipecahkan menjadi 2 pyruvate. Selain daripada tu, dia akan menghasilkan 2 NADH dan 2 ATP. Okay. proses glycolysis ni dia berlaku dekat cytoplasm. So, macam yang kita dah belajar before ni, yang kita fokuskan kalau ada kehadiran oksigen kan. So, kalau adanya kehadiran oksigen ni, pyruvate ni akan terus melalui proses link reaction dekat mitokondria ni untuk menghasilkan acetyl-CoA. So, bila dah hasilkan acetyl-CoA, dia akan teruskan proses Krebs cycle dekat mitochondria matrix. So, dekat proses Krebs cycle ni lah akan adanya banyak production of NADH dan NADH2. So, bila dah banyak sangat NADH dengan NADH2 ni, baru dia akan pergi dekat electron transport chain ni, dekat kristal ni, untuk menghasilkan 38 ATP. Gitulah. Okay. Tapi itu kalau adanya oksigen. Untuk proses fermentation ni, dia does not require oksigen. Maksudnya kat sini, kalau body kita kekurangan, lack of oxygen, so instead of dia melakukan proses aerobic respiration ni, dia akan go to sini lah. Dia pyruvate tu dia akan stay dekat cytoplasm, dia tak akan masuk ke mitochondria. So bila dia dah dia tak ada oksigen and dia tak melakukan aerobic respiration, that's why akan berlakunya proses fermentation. So fermentation ni yang korang kena tahu, dia ada dua types. Satu, alcohol fermentation, dia berlaku dekat plants, bacteria, yeast. And lagi satu, dia lactate fermentation, iaitu berlaku kat animal, termasuk manusia lah. Kita pun animal kan. So boleh dikatakan yang proses fermentation ni ialah anaerobic respiration, iaitu tanpa kehadiran oksigen. Macam yang Alda cakap tadi, proses glycolysis tu to oxidize glucose into pyruvate. Proses ni sebenarnya, fermentation ni sebenarnya dia nak sustain glycolysis proses. Dia nak kekalkan proses glycolysis ni supaya lebih banyak penghasilan NADH2 tanpa adanya aerobic respiration. Penerangan dia macam ni. In the absence of oxygen, pyruvate cannot be transported into mitochondria to be oxidized further. Maksud dia, dia tak akan melalui yang aerobic respiration tu, yang ada Krebs cycle, link reaction tu semua kan. So, bila tak berlakunya proses aerobic respiration ni, pyruvate tu akan remains dekat cytoplasm. So, kalau dia dah remain dekat cytoplasm, of course lah penghasilan ATP tu akan berkurangan. So, kehadiran oksigen tu lah yang akan menentukan, dia akan tentukan either kita nak buat proses aerobik atau anaerobik. So, untuk case fermentation ni, dia kan tak memerlukan oksigen. So, let's say yang kalau body kita lack of oksigen, body kita akan melakukan proses fermentation. Then, as glycolysis continue, the concentration of NADH increase, the concentration of NAD plus decrease. Macam yang kita dah tengok dekat chemical equation ni, nampak tu, proses glycolysis ni je dia dah menghasilkan 2 NADH. So, kalau pyruvate ni dia tak melakukan proses aerobic respiration, penghasilan NADH ni akan meningkat dan akan banyak. Bila dah banyak, dia tak ada proses apa-apa yang lepas tu. So that's why dia kata NAD plus tu akan berkurangan, dia akan decrease. So bila dah berlakunya macam ni, sel kita akan menghadapi limited supply of NAD plus. Bila dah berlaku sangat macam ni, proses glycolysis dia takkan berlaku sebab tak ada NAD plus yang menolong dia untuk menghasilkan pyruvate. So, in order to sustain glycolysis, NADH must transfer its electron to another organic compound. Barulah bila NADH ni dia dah ditransferkan kepada organik compound yang lain, then barulah NADH ni akan dioxidize. So NAD plus is regenerated. Okay, two types of fermentation. Satu ialah lactate fermentation, iaitu pyruvate direduce menjadi lactate. Yang kedua, alcohol fermentation, acetaldehyde direduce menjadi ethanol. Okay, so kita sekarang masuk dekat lactate fermentation. Ni glycolysis tadi, glycolysis. So dua pyruvate yang ni dia lah yang akan direduce untuk menjadi lactate dengan adanya dua NADH. Okay, ni lactate fermentation ni occurs in animal cell during strenuous activities. Strenuous activity ni dia macam activity berat. Activity berat macam kalau kita jogging, kita bersenam, kita berlari, kan? Kita memerlukan oksigen yang banyak, kan? Untuk inhale and exhale. So, when O2 is insufficient, dia berkurang untuk support aerobic respiration ni. That's why berlakunya fermentation ni. When O2 is insufficient to support aerobic respiration, glycolysis occurs to oxidize glucose into pyruvate. Ni cerita dia. Bila dia kekurangan oksigen, pyruvate ni dia receive electrons for NADH yang ada banyak-banyak terhasil dari proses glycolysis tadi. So bila dia dah receive electrons from NADH, causing it to reduce to form lactate. Okay, NADH is oxidized to regenerate NAD plus to sustain glycolysis. How dia boleh jadi lactate? Sebab dengan adanya NADH dengan 2H plus ni, so dia akan di-reduce untuk menjadi lactate. So macam yang kalau korang perasan, kalau korang lepas bersukan tu, korang akan rasa sakit-sakit badan, korang akan rasa lenguh-lenguh. Sebab adanya lactate tu dia berkumpul dekat korang punya otot. Sebab tu korang rasa penat sangat, korang rasa lemah. Bila korang dah rasa macam tu, of course korang need oksigen, kan, untuk proses inhale, exhale korang. Oksigen yang korang tarik nafas, korang masukkan oksigen dalam body korang tu, itu sebenarnya untuk memecahkan lactate tu menjadi carbon dioxide plus water. Next, for alcohol fermentation, sama juga dari glycolysis, cuma bezanya nanti dia akan melalui proses decarbonylation, iaitu pembuangan CO2 untuk menghasilkan dua acetaldehyde. And then macam biasa, dua acetaldehyde ni dia akan accept 2H plus and dua elektron gitu. 2 elektron untuk di-reduce menjadi 2 ethanol. Bila adanya NAD plus ni, then proses glycolysis sustain lah, akan kekal. Itu je lah pusing-pusing dia. Macam yang Al dah cakap tadi, pyruvate akan undergo decarbonylation, iaitu proses removal of carbon dioxide. Pyruvate undergo decarbonylation to be converted into acetaldehyde. Then acetaldehyde itulah yang akan receive electrons from NADH, then causing it to reduce to form ethanol. Then NADH is oxidized to regenerate NAD plus. Ni glycolysis. So pyruvate yang terhasil ni dia akan undergo decarbonylation untuk pembuangan CO2. Dua pyruvate, kan, so dua CO2. So bila dah buang CO2 yang dia buang tu akan terhasilnya dua acetaldehyde. Then dua acetaldehyde ni lah yang akan di-reduce untuk menghasilkan dua ethanol. So 2H2 plus dengan 2 elektron akan digunakan oleh acetaldehyde ni direduce menjadi 2 etanol. So bila 2H+ ni dah diberi ke acetaldehyde, dia akan jadi NAD+. Okay, 2 NAD+ dah hasil, dan NAD+ ni boleh sustain glycolysis. Sebab tu dia macam cycle. Itu. I think that's all from me. Thank you.